Good morning. Today I'll be talking about some work we've done looking at the prognostic value of CD68 positive macrophages within cholangiocarcinoma tumors and how the value of these cells depends on their location within the tumors. First of all, I'd like to thank the AMMF for giving me the opportunity to present my data to you all. It's a great honor. So I'll quickly talk through my outline. I'm going to talk about why we're interested in looking at cholangiocarcinoma tumors in the first place. I'll talk about the methods we use for analysis. I'll take you through some of our results, and I'll end by concluding the data, summarizing the data, and talking about where we think this study can go in the future and why we think it's important. So, as an introduction, we know that cholangiocarcinoma is the second most common form of liver cancer in the world, and it arises from a malignant transformation of cholangiocytes, which are the epithelial cells lining the bile ducts within the liver. Rising incidence and mortality rates, combined with a very poor prognosis of 5-year survival less than 10%, make this a really important clinical problem that needs to be addressed. Now, previous studies have identified some recognized risk factors for cholangiocarcinoma, which include infections like chronic hepatitis B and C, infestation with the liver fluke in Thailand, and in the UK or in the Western world, it's also linked to autoimmune related diseases such as primary sclerosing cholangitis. Now, all these risk factors point to an immune component in the pathogenesis of cholangiocarcinoma. However, immune related mechanisms of disease pathogenesis and progression remain largely understudied. And a better understanding of the immune microenvironment in this disease is really necessary to enhance the development of new treatments and prognostic strategies for patients with this terrible cancer. So we therefore wanted to investigate the potential contribution of immune cells to cholangiocarcinoma pathology. So how did we do this? Well, first of all, we asked the question, can we identify tumour-infiltrating immune cells within cholangiocarcinoma tumors, and will there be a difference when we look at tumorous bile ducts compared to non-tumorous bile ducts from the same patients? So to look at this, we used immunohistochemistry on formalin-fixed paraffin-embedded tumors. We looked at about 20 tumor samples to investigate the presence of these cells. After that, we then went on to ask, can we find a significance, or is the presence of these cells significant in clinical outcome? And to do this, we undertook a systematic review and meta-analysis of existing literature in accordance with the PRISMA guidelines, which I've referenced here in reference 1, so we can look at it in more detail if we're interested. And we did this to determine the correlation between immune cell presence assessed histologically and clinical outcomes in patients with cholangiocarcinoma. I'm just going to skip through to the next slide where we've summarized our systematic review and meta-analysis protocol. We can see it on the slide. So this schematic just takes us through the steps we took to carry out this protocol, the identification of papers, screening, assessment of papers for eligibility, and the final inclusion of the studies. And on the left, you can see the steps we took to ensure that our study was robust and reproducible. And we have registered this study on the Prospero, the International Prospective Register of Systematic Reviews. So please feel free to have a look at that for more information on what we've done. Finally, after looking through after carrying out our systematic review and meta-analysis, we used nanostring digital spatial profiling technology to preliminary undertake spatially resolved analysis of immune protein signatures within 2 cholangio tumors. So I'll touch again on the methods we've used as I talk through the results. So what did we find? First of all, from our immunohistochemistry, we carried out staining for different immune cell subsets. In figure 2, we're showing staining for CD68 positive macrophages, and these are identified as the brown dots within these tumor sections. So on the top, and on the left-hand corner, figures A and C, these represent samples from tumor, and on the right hand, figures B and D, samples from non-tumor from the same patient, so non-tumor bile duct tissue. And we found that we could very clearly identify infiltrating CD68 positive macrophages within the tumor stroma, but also within the tumor islets, which I've delineated by these with this black line drawn around the tumor islets. And if we look at a comparable, if we look on the right in the non-tumor tissue, we can see comparatively very few brown dots surrounding the non-tumorous bile duct. Now, if we go down to the lower panel, what the lower panel represents a zoomed in image of the figures shown on the top panel. So in the lower panel to the left, C, we can see the bile ducts indicated by these black arrows, and we can very clearly see a large number of infiltrating CD68 positive macrophages, I beg your pardon, surrounding the tumorous bile ducts. And on the right hand, on the lower right, in figure D, we can see the bile duct here, identified by its placement next to the hepatic artery and hepatic vein, also known as, together they're known as the hepatic triad. We can see comparatively fewer brown dots representing CD68 positive macrophages. And when we quantified this, we found that we had statistically significantly more macrophages within the tumors compared to the non-tumor. Excuse me. So we also, like I said before, we also looked at other immune cell populations. So in figure 3, on the top left, we looked at CD3 positive, which represents the total T lymphocytes, CD3, CD4 double positive, which represent the helper T lymphocytes, and these are a subset of the total lymphocyte population. CD3 in the lower panel, CD3, CD68 double positive, which are the cytotoxic T lymphocytes, and lower right panel, CD20 positive B lymphocytes. And what we could see clearly that there were significantly more CD3 total and helper T cells within the tumor compared to non-tumor. Within the cytotoxic population on the lower right panel, on the lower left panel, sorry, here, there appeared to be a trend towards greater cytotoxic T lymphocytes within the cholangio tumors compared to non-tumor, but that didn't reach statistical significance. And when we look at B cells, on the lower right, we can see that there was no difference between infiltrating B lymphocytes within cholangiocarcinoma tumors and non-cholangiocarcinoma tumor bile ducts. Interestingly, what we found was in all the samples we assessed, there were very few B cells to be seen, thus indicating that potentially B cells don't have that much of a role or are not represented, rather, within the tumor microenvironment. So what have we found? We found that tumor-infiltrating cells are enriched within the tumors compared to non-tumor bile ducts. The next question we asked was, are these cells of clinical significance? And this was why we did the systematic review and meta-analysis. So what we did was to assess different studies which evaluated the presence of tumor-infiltrating lymphocytes within different cholangiocarcinoma tumors, and we selected only studies that assess the presence of infiltrating lymphocytes histologically, because we wanted this to be the baseline. Should I use the word comparator? That we wanted this to be the baseline for all the papers we assessed. And we found something really interesting in terms of the presence of CD68 positive macrophages. I should say that at the end of our study, we did find that the presence of CD68 macrophages and CD3, CD8 positive cytotoxic T cells were the most significantly associated with overall patient survival in patients diagnosed with cholangiocarcinoma. But on further investigation, we found really striking data in terms of the CD68 macrophage presence, and that's why we've chosen to show this for this poster. So what did we find? We found that if we split the tumor into different histological regions, or different regions, let me say, so assessing the tumor invasive front versus the center of the tumor, we got two very different results in terms of the significance of infiltrating macrophages with overall survival. And again, our results again changed when we looked at papers where they assessed tumor as a whole. So figure 4 is, these are forest plots, which is showing the correlation between intratumoral location of CD68 macrophages and overall patient survival. Now, if we take this central line as our center, and that's represented by the number 1, everything to the left of the line represents increased overall survival in tumors bearing CD68 positive macrophages, and everything to the right represents decreased overall survival. So let's start at the top. For the top panel, these papers looked at the presence of macrophages in the tumor invasive front, also known as the leading edge or the pushing edge of the tumor. We had about, I think, 3 papers with a total of 174 patients, followed up for a maximum period of 250 months. And we can see that if we focus on the diamond, we can see that the diamond is to the left of our central line, and the diamond is quite spread out. And this indicates, or the interpretation of this is that this paper found that if you have macrophages in the tumor invasive front, then these patients have better survival, so they live longer. Now we go down to the middle panel, where these particular papers looked at the center of the tumor, and there were 2 studies with a total of 127 patients recruited, and these patients were followed up for a maximum of 250 months. Now, again, taking this line as our center, we can see that the diamond is to the right of the line. And the interpretation of that is that these papers found if you have patients with CD68 positive macrophages within their tumors, then these patients died earlier, so they had decreased overall survival. And the lowermost panel, we have 2 papers where they looked at the tumor as a whole. They didn't divide it into either they didn't look at the invasive front or the tumor center, they just looked at the entire tumor. And these studies recruited a total number of 361 patients who were followed up for a maximum of 125 months. And we can see, again, using this line as our center, that the diamond is really close to the line and also is quite narrow. It's a lot less broad than the top 2 diamonds. Now, the interpretation of that is that these studies found if you assess the tumor as a whole, there is no effect of if you assess the tumor as a whole, the presence of CD68 macrophages has absolutely no effect on whether these patients survive longer or not. So this last panel shows that CD68 has no effect. So very differing results depending on the location within the tumor wherein these macrophages were assessed. Now, finally, we thought to ourselves, okay, this is all well and good, but can we look at spatially resolved analysis of macrophages and indeed other immune cells within tumor samples? Is that something we can possibly do. So this last bit was done in collaboration with, the Cancer Research UK Centre at Birmingham and Nanostring Technologies in Seattle in the US, and we're really grateful for their collaboration. So what we did is we stained cholangiocarcinoma tumors. So the staining is shown in the top left panel. So these are that's figure 5A and B. And these tumors were stained to identify tumor cells, which are seen in green, and green represents pancytokeratin. It was stained for T cells, which are the red dots. Those represent CD3 positive T cells. Stained for stromal cells, and that's the yellow stain, which represents smooth muscle actin, and the cell nuclei were identified by DAPI staining, which is seen in blue. And figures A and B represent are images of the tumor obtained from a patient of UK origin. And after staining, we selected regions of interest, abbreviated as ROIs, and these are represented by the white boxes or the circles seen on figure A here. And these regions of interest represent either tumor-rich, immune-rich, or stroma-rich areas. Excuse me. Excuse me. And these regions represent, as I said, they represent either tumor-rich, immune-rich, or stroma-rich areas. And figure B just shows representative examples of what I mean by a tumor-rich area or an immune-rich area. So, for instance, the first box on the topmost panel represents tumor ducts surrounded by infiltrating CD3 positive T cells. The second box on the lower panel represents a mix of immune cells and stromal cells. So this just gives, this is just an example of what the different regions looked like. And then based on these regions of interest, using the nanostring technology, we were then able to further probe within these specified regions of interest to generate quantification graphs and heat maps of different stromal and immune proteins in our bid to look for a particular stromal or immune signature. So figure C is the data obtained from a patient of UK origin, and figure D is the data obtained from a patient of Thai origin. So a patient from Thailand. So if we focus on the left panels in figures C and D, what we did here was to quantify the presence of CD68 positive macrophages in the different regions of interest. So the labeling at the bottom represents the different regions of interest, ROI 1 through 12, I think, in the UK tumor, and ROIs 1 to 7 in the Thai tumor. And we can see that within these different regions of interest, graphically represented, we can see that we have very different numbers of CD68 positive macrophages. So again, this ties into what the systematic review found, that potentially you have differential presence within different areas of the tumor. And if we look at the heat maps on the right-hand side of panels C and D, we can again see in a more colorful representation, let me say, that you have differential expression, or differential presence rather, of CD68 positive macrophages within the distinct tumor regions delineated by the regions of interest. Again, regions of interest 1 to 12 in the UK tumor, and I think 1 to 7 in the Thai tumor. Interestingly, we also found that within both the UK and Thai tumors, CD68 positive macrophages clustered with the immune checkpoint molecule B7H3, which we thought was a really interesting find, showing that the pattern of immune cell clustering was fairly similar within these two tumors. Of course, this is preliminary work that we've done just on 2 patients, like I said before. So it's an interesting pattern to look at for in the future, but we're not going to draw any conclusions from that presence, from this data just now. So what have we shown? What has our study found? We found that CD68 positive macrophages and CD3 positive T cells are enriched within cholangiocarcinoma tumors compared to non-tumor tissue. We found that CD68 positive macrophages, which infiltrate into the tumor, correlates with overall survival in patients diagnosed with cholangiocarcinoma. We found that the prognostic significance of tumor-infiltrating macrophages, tumor-infiltrating CD68 positive macrophages, appears to depend upon the location within the cholangiocarcinoma tumor. And we think this is likely related to the underlying intra and intertumoral heterogeneity in terms of the biology of the tumor, but also in terms of the tumor microenvironment. So for example, within the center of the tumor, we know that the centers of tumors are usually very necrotic and have a high amount of hypoxia compared to the invasive edge of the tumor. So this is just an example of a biological difference that could potentially underlie this difference we found. And finally, I think these findings highlight the need to consider spatial context in our evaluation of tumor specimens in research and indeed even in diagnostic or prognostic endeavors when we're assessing cholangiocarcinoma tumors. So I'd like to end by again thanking the AMMF for giving me the opportunity to present my data. I'd like to thank all my collaborators and supervisors on this project. I'd like to thank the University of Nottingham, where I'm hosted, and my funders, the National Institute for Health Research, who fund my clinical lectureship position, and the Pathological Society of Great Britain and Northern Ireland, who funded part of the work. Again, I'm very grateful to NanoString Technologies for giving us access to their digital spatial profiling technology. And thank you for your attention.